Hello and welcome to the Microscopes revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. Magnification is how many times bigger the image is compared to the original object. Resolution is how clearly 2 points can be distinguished as separate points. Light microscopes were first used in the 1600s. These used light and glass lenses to form an image. It allowed scientists to see and draw cells for the first time. They were limited in detail because of lower magnification, a maximum of about 2000 times, and a lower resolution. The lower resolution is due to a property of light rather than the microscope design. Electron microscopes, which were developed in the 1930s, use beams of electrons instead of light, and there's two main types. There's a transmission electron microscope, TEMs, and a scanning electron microscope, SEMs. Both of these have much higher magnification, 2 million times, and a much higher resolution, so they can see very fine details much more clearly. With greater magnification and resolution, electron microscopes can reveal the detailed structures within cells. This allowed scientists to now see the internal structure of mitochondria and chloroplasts. They could also view ribosomes and viruses, which were not previously visible with light microscopes. For magnification calculations, you will need to convert units, so that both of the numbers that you use in the formula that's on the following slide will have the same units. So if you need to convert centimeters into micrometers, first of all you will times the number of centimeters by 10 to turn it into millimeters, and then the millimeters you will times by 1000 to turn that into micrometers. For magnification calculations, the formula we use is that magnification is equal to the size of the image divided by the size of the real object. And we've put down the formulae triangle and the three possible rearrangements. An animal cell has a diameter of 30 micrometers. When the diameter of the cell was measured using a ruler on a piece of paper, it was 3 centimeters. Calculate the magnification. So the real size is 30 micrometers. The image size is 3 centimeters, because that is what has been drawn on the piece of paper. We need to convert them into the same units. So 3 centimeters is 30 millimeters or 30,000 micrometers. We will use the formula magnification equals the size of image over the real object size, and so the magnification is 30,000 divided by 30, which is times 1000 for magnification. Thank you for watching. See our revision notes, the link is in the description.